session is co-sponsored by the Center for Animals and Public Policy and the American Society of Lab Animal Practitioners student chapter. I want to especially thank Ginny for knitting the electrons together throughout the whole continent. Yes. And also Colleen, who is the president, Colleen Thurman, president of ACE Lab, for reaching out and inviting Dan. Especially thank you to Dan for agreeing to share your research with us. Dan is the professor of the Animal Welfare Program and the Associate Dean of Graduate Studies at the University of British Columbia. He has the Industrial Research Chair in Animal Welfare, Natural Sciences and Engineering from the Research Council of Canada. He's a founder of the Animal Welfare Program in British Columbia in 1997. Much of his work has been with livestock and large animals, but has also done some innovative work with improved housing for laboratory rodents, which is what we're going to talk about today. In addition to an almost full lecture hall here in Grafton, there's half a dozen people participating via the web throughout Boston, and then Dan is comfortably in his office in British Columbia, where it hopefully is warmer than it is in Boston today. So I'm going to turn this over to Dan. At the end, I'm hoping there's time for questions, and in order for everyone throughout the continent to hear them, if you have questions, you can either come up to the microphone and use the microphone, or put them on a piece of paper and I'll read them out. Okay, Dan, thanks again.
Okay, well, thanks very much for the kind invitation to be able to speak to you today. I'm sorry I can't be there in person, but thanks especially to Ginny for her efforts in setting up everything on the tech side. So I'm going to sort of walk you through a story that has been sort of unfolding in our lab over the past few years, and it really reflects maybe a broader change in animal welfare science, where when the field started off, we were very interested in addressing sort of acute conditions of suffering, and the conversation has changed a little bit to ask more questions about whether or not we're providing a good quality, overall good quality of life to the animal. That includes assessment of positive states and allowing the animals to do things that they're highly motivated to do. This talk is very much a collaborative effort with two close colleagues and students, Joanna, who's been doing her PhD in the program, who's done some very innovative work in terms of, as you'll see in a minute, sort of these radically reimagined caging for rats, and Joyce Reinhold, who really brought to our lab a much better understanding of the importance of socialization and the very important role of human handlers in terms of improving the lives of the animals we work with, and including in this case rats. When I started preparing for this presentation, I asked my colleagues in doing rodent work in different parts of the world what they thought, sort of what would be the ideal environment that we could keep rodents in, and sort of what would be our rodent utopia. And in one conversation I had with Joe Gardner, who's now at Stanford, Joe said, he thought for a minute and he said, well, I'm not sure I can tell you what rodent utopia is, but I've got to guess what rodent dystopia would be. And that would be being in an environment that's much brighter than you prefer, quite a bit colder than you prefer, but you don't have access to the right materials to build proper shelter for yourself, where you're kept up high, where you have these sort of looming potential predators come over you from time to time. And in short, basically, he described what looks awful lot like some of our current rodent rooms, and that maybe all those things together can have profound effects, not only in terms of the lives that those animals experience, but also in terms of the way we see those animals. And so here is one of our rats in a conventional cage. And this looks a bit boring because it's a still picture, but what I'm going to do is just pause with me for a second while I master the technology here, but I'm going to try to bring up the video at the same time. Just give me 1 second here. And here's this video, okay? Just to show you what this looks like. So when you play the video, and this is what the rat looks like to us then, as we watch it. And in short, not very interesting, right? So here's 2 rats. Oh, there you go. You saw the head move. But this gives us a sense of what these, when we keep animals in this kind of environment, what they look like to us. Okay, so keep that in mind. Let's go back to the presentation for a second. So I'm going to introduce you to the 2 stars of the show. Here's Joanna and Joyce, and walk you through some of the things that they've been working on over the past couple of years. So here is one of the structures that Joanna's created. And what this is caging, it actually is a commercially available cage that the rat fanciers, people who keep rats for pets, use. And Joanna set it up using sort of a combination of tricks that she picked up, including from people who keep rats as pets. And a lot of these sort of devices are easily washable and things which she could get cheaply. There's a climbing structure, things for the animals to hide in, etc. A lot of vertical space. You can see the animals can go up and down different levels. And at the bottom here, Joanna's created an area where the animals can also burrow, and I'll show you more of that behavior in a minute. In Joanna's work, she used sprag dollies, which she obtained after weaning, and in all her work, she's keeping animals in a reverse light cycle so that our active time corresponds with their active time, to allow us to see them, but so as to not disturb the animals. We're using these low pressure sodium lamps, which work really well in rodent rooms. It's a color that the animals themselves are basically blind to, but we can see quite well in, and that our video cameras can see very well in. But you're going to see as we go along that all of our videos have sort of a little bit of a yellowy tinge, and so that's why. So this is an example of how Joanna had her room set up, and you can see a bunch of different cages on this side, and there's similar cages on the other side, and also interspersed are a bunch of standard housing so that she could look at the development of the behaviors and the behavioral repertoires of these animals in the 2 different conditions. So just to, here is now Joyce's setup, which is very similar cage, in fact the same commercial caging, but she just set it up in a couple different ways. She wanted actually something which was a little bit easier to maintain. So instead of using soil, she's used these fleece lining. And you'll see that the animals also burrow in this layers of fleece in quite interesting ways. It's sort of another interesting setup. But it basically is just to show you that it probably doesn't, the details of exactly how it's set up, from our perspective anyways, in our experience, probably don't matter so much. Joyce was using Long Evans rats, and she'd also obtained them after weaning, and she's also keeping them on this reverse light cycle, as I explained. Okay, let's have a look at what this looks like now. Can you guys see that video on that end? I'm hoping you can. Just shout if you can't see that video. Okay, so 2 things I want you to notice is just look at the different things that the animals are doing. This is actually sped up. It's a 4 times speed. And the thing I want you to realize is that at the same time that video is playing here, we'll play this video at the same time. And so these are same rats, same time, same environment. And you can look at the diversity of behavior of the animals in the one setup in relation to the other setup. There's a bunch of different things going on, but you can see that it's quite easy to see at least the differences in terms of the amount of behavior and the diversity of the behavior that the animals are showing. So there's this animal actually moved its head. There you go. I'm moving on this side, but contrast the 2 environments again. Okay. So let's just walk through some of those behaviors that we see in the enriched environments that Joanna and Joyce have set up. And one of the key ones here is this climbing behavior. Obviously, these cages provide all the sort of opportunities for moving in the vertical axis. Here you see a still of animals in the 2 different environments, and we'll just set that up, and we'll see if the video runs on this side. If it doesn't, I'll switch over to the other side. No, it doesn't. So just give me a second, and I'll show you a very cool video of this bounding behavior. So here, open this up. And here we go. So you can see some of the these jumping movements that these animals are able to do. That's in Joanna's cage, you can see. That's over to Joyce's cage now. And look at them doing these big jumping movements between structures in their cages. This is slowed down now at 1/4 speed. So just to give you a better sense of what the behaviors are and the sort of athleticism of these animals kept in this environment. Just quit out of that. Oh, and then go back to oops, not that one, the climbing one, which is the one I wanted to show you. So here's our rats. Just open that up big so you can see it nicely. Just some of the climbing movements. Very good at making their way across the cage on the walls. These again, now over to Joyce's rats, and you can see these animals doing a little bit of a monkey bar movement here. There's another individual sort of skirting across on the face of the cage. Okay, cool. Let's close out of that. And 1 second here. Go back into this. Another important behavior that you see is this perching. You might have noticed that on the previous videos I showed, that the animals use the space not only for climbing on, but they actually like to get up high and sort of like Yergul the turtle and survey everything that's beneath them. So here you can see one of the animals sitting up on a bit of one of these PVC pipes, checking the world out. There's another one using the hammock, not as a place to lie on, but a place to perch on. And here's this sort of climbing structure here, which is actually a IKEA sort of $1.98 bag holder that Joannas repurposed for the rat enrichment. And you can see again the rats using it as a way of getting up high and surveying their world. Another interesting behavior is this full rearing. So rats like to get up on their back feet and stretch, and this is very easy to do in these cages with lots of vertical space. Here I want you to see, so here's one of Joanna's rats in that soil substrate, so it's down at the bottom, and you can see clearly they're able to rear up. You can see also the way Joanna set this up, she actually has a bit of clear plastic, so we can see the burrowing structures that the rats make. I'll show you that better in a second. But what's important here is you see the rat rearing fully up on its legs. And then you look at our standard housing, you see the animals aren't able to do this. I mean, they're able to get part way up, but they're not actually able to fully rear in our standard housing. The bounding and running, so I showed you that video already of those animals in slow motion and sped up, sort of doing these big athletic jumping movements around the cage, and that's clearly something they're able to do in these larger cages. So I won't show you that video now because you've already seen that. They obviously spend a lot of time interacting with the various enrichment devices we provide for them. Here they are sort of hanging out in the hammocks, and makes for very cute pictures, obviously. And here's some other animals hanging out in Joanna's IKEA plastic bag recycling structure, and again, works very well. Here's another picture of that. Okay, let's talk a little bit about the burrowing. So as I said, Joanna she was very interested in seeing what the rats would do if you gave them the opportunity. They use this a lot. They tend to remake their burrows almost every day. And they'll take things down and bury them in there. And so there's a lot of activity associated with substrate if you provide it for them. There's an animal emerging over here on this one. Let me show you a video of them using that burrowing structure now. Just give me a second. And we'll open this up. Okay, and here you can see is one animal down there digging. Another animal's tail just disappeared up here, so he's digging somewhere else. But this one's giving us a show because he's right up against the side. There's another animal just emerging from a burrow over there. And here we see them in Joyce's apparatus. As I said, seem to be using these layers of layers of fleece to use that like a burrowing structure. So you can see these sort of random bouncing movements underneath the blankets here are basically rats scurrying back and forth and using the different layers of that substrate. Okay, we'll quit out of that burrowing video now, and we'll go back in the main presentation. Okay, one of the things that both Joanna and Joyce were very interested in was in not just adding sort of furnishings to a cage that you leave there forever, but also finding ways of mixing things up. So having something which is interactive for the animal, and also something which can be varied on a day-to-day basis by animal handlers, depending on what they have available in their own interest. And here's one option that Joanna did. She just basically took old toilet paper rolls and she would fold some hay into them, and that seemed to entertain the animals because they could destroy the toilet paper roll, which was great fun, of course, and they could also tear out the various bits of hay and chew on that too. There's the other treats that she could put into here on a day-to-day basis, depending on what she had. Here's another option that Joyce and Joanna used, which is basically just a cat ball. And you can see the cool thing about this feature, this little piece of plastic, is you can rotate the two spheres. And when you do that, you can change the size of the aperture, so more or less treats come out. And the animals then basically earn their treats by pushing this ball along, and every time it rolls, maybe a treat or two will fall out on the side. So let's have a look at what that's like in action. I'll just quit out of the PowerPoint and show you the treat ball video. Just give me 1 second. So here actually are 3 of Joanna's rats, and they're actually down in the soil substrate, and they're working away at one of these treat balls, as you can see here. And here they are. They've got the treat ball up in the upper part of the cage here. So very simple little device, but it certainly gets the animals working and interested. Okay, we'll quit out of that treat ball video. We'll go back into the PowerPoint. Okay. So just sort of summarizing then the various things that we've seen so far. We're going to go through the kinds of behaviors that we can see in the semi-naturalistic housing that Joyce and Joanna were using versus what we see in our standard housing. And I mean, obviously, one thing that we can provide in this environment, especially in Joanna's case, is this opportunity to dig and burrow. But I showed you how Joyce, using a different type of setup, also allows the animals to show some of the same behaviors. We don't know whether that's equally satisfying to the animal or not, but it gives you an example about what a creative caretaker can do when they're interested in providing an environment which allows the animal to express the behavior. They don't always have to use the same tools to achieve the same outcomes. Climbing, I mean, obviously we're creating a structure here which facilitates that, which actually encourages it because we have this vertical aspect to the cage, and the animals do a lot of it. They actually seem to be quite athletic in that kind of environment. The animals use the vertical space also, as I said, to perch. They like to get up high and look down what's below them. A very simple behavior, but interesting one, is just this rearing. The animals are able to stand fully up on their back legs and stretch out, and that's, again, something which is not possible to do in standard housing. There's these bounding running movements, which obviously just require enough space for the animals to do it. And you get the animals doing these things in the environments I've shown you, and they're simply impossible to do in a standard housing. Object interaction is something that we chose to provide here. Of course, could also be provided in our standard caging, but often isn't. But it's certainly something I think is a sort of simple way that a lot of people working with these animals could start in terms of making that environment more interesting for the animal. And also, something we'll come back to again and again, is this idea of also showing us a little bit more about the complexity and sophistication of these animals. I think that's, to me, I think that's one of the most sobering aspects of this work, is just seeing how neat these animals are when you give them the opportunity to sort of show their ratness. Okay, I've showed you sort of these commercially available but sort of rat fancier cages. If you show these to your average manager of a rodent unit, they'll probably have a heart attack because they're very unlike the systems they're used to working with. So I wanted to also walk you through some other kinds of options which, for people that are interested in trying something a little different, are available. So here's a cool setup that some researchers in Colombia have been using. This was some videos given to me by Becca Franks, who's now a postdoc in our lab, but was doing this work in Colombia. I'll just show you what her, in this case, mouse cages look like. So she basically took the standard mouse cages and she just provided the opportunity for the mice to travel between them. You can see here's a short tube, just go between these cages. Here's a longer tube, they can go between these cages. And it basically, I'll just go back to the previous slide because it doesn't show it in that video. Also, one of the cool things she's able to do is provide an opportunity where the animals can access a darkened cage. And as we know, these animals, these rodents, of course, normally prefer to be in a dark environment. So giving them a choice to escape to a darkened area might be important. Here's just a close-up of some of these animals interacting with some of those kinds of structures. So they certainly use it and take advantage of that kind of complexity when you provide it for them. Here's a commercially available caging system, the Marlowe cage, which again provides a little bit more opportunity for the animals to do sort of some interesting things. Actually, you can see down at the bottom, the animals have running wheels and can access food and water. They can enter these areas via access to a maze, and that maze can be changed around, which can be interesting from a researcher's perspective, but also from the perspective of just adding a bit of cognitive complexity for these animals and just changing things up on a day-to-day basis. Here's just an example showing the various different setups that this one group of researchers did in terms of the maze structures. So some opportunities to do interesting things. And one of the things that these researchers found is that just providing that opportunity to this enriched environments had quite important differences in terms of the effects of on those animals. And so one of the things they did is they looked at how those animals responded in a water exploration, a water maze test, and they found that the conventional reared animals did this sort of wall following behavior. You can see the dark red line showing what all the animals did. Well, the animals reared in the enriched environments actually showed a much more complex patterning. And this complex patterning, of course, meant that the animals were much more likely to be successful in terms of finding the area in the middle where they could stand upon. Here, just as a simpler option still, this is a commercially available caging system, which just provides a higher lid. And one of the advantages you can see there is the animals can do this full rearing behavior. So there's one positive. There's also the opportunity to add perches. So you can here you see an animal actually using that perch up there. And there's also the opportunity to use slightly different types of enrichment devices inside the cage because you just created more vertical space or more a total volume in that cage. Okay, so we talked a little bit about the physical environment. What I'd like to do is now just go back to talking about which may be the most important aspect for many of the animals we work with, certainly animals we work with intensively, and that is the social environment we provide for them. And that social environment can, of course, be other cage mates, but often it can also be the human handler. And I think in many of the projects we do, the handling ends up being, you know, at best, a neutral kind of thing for the animals, and often something that's quite aversive. The handling is associated with, you know, being picked up in a way that's maybe not pleasant for the animal, being restrained in a way, maybe even only for weighing or for some kind of sampling, and so that we certainly, I think, are not taking full advantage of what we can be in terms of turning our interactions with the animal into a positive thing for the animal. And again, I'll come back to this idea that if we could find ways of doing that, I think it would also then much improve the level of satisfaction that we have as researchers and as animal technicians in terms of our own interactions with the animals. I think most of us that go into these careers working with animals do it because we love the animals and we're actually looking for opportunities to do interesting things with them. And I think that we need to have a little bit of a reboot, a rethink in terms of the very fundamental ways we go about in terms of interacting with laboratory rodents in particular, in terms of making those interactions much more pleasant and reinforcing for the animals. And I think that in turn has the opportunity at least to dramatically change the amount of satisfaction that we as researchers and animal technicians and animal veterinarians have in terms of working with those animals. So, of course, in a lot of cases, animals must interact with humans, and if we pick the right types of procedures, we may be able to at least reduce the aversion and anxiety associated with that. And I'm going to show you a really nice example from Hurst and West, and maybe some of you guys know this already, but it just shows an example how in this talk I talk a lot about sort of blue sky stuff. Utopian type vision for where we could go in terms of laboratory, our interactions with laboratory animals. But I also wanted to point out some sort of simple places where we can start. And I think one really simple way is just how we pick up these animals. So I'm going to show you some examples from this Hurst and West group, which I think are really cool. And so you can here see the animal, a mice being picked up by the tail. And now just let me show you some videos of what they did. So here you have, on this first video here, is the researchers interacting with mice in the very first time when they're picking them up by the tail. I'm going to start the video and I'll show you what they're doing. They just go and they grab the mouse, and they just hold the mouse gently there on the researcher's forearm for a period of time. And then when they did it, they went back and did this day after day after day, right? Okay, quit out of that. So, close that. Now, let's look at the animals. This is a different set of animals, but here's the same researchers that have been trained that have done now multiple tests. I think this is the 4th or 5th day that the animals have been exposed to this tail handling procedure. And now, instead of actually going to pick the animals up, the researchers just stick their hand in the cage. And look at what these mice do. You see they actually do a bit of protective bearing on the hand. They certainly don't want to go anywhere near that hand that is now that they've now learned to associate as something that comes and picks them up. Okay. Now, let's look. I'll close that tube. I'll close that tail one. I'll go to the first. This is another sort of technology that can be applied to the same issue, and that's picking the animals up just with a clear tube. So I'm going to start the video so you can see what I mean. Here's just a two-sided tube, and they just gently pick the animal up. This is the first time the animal's in it. And you can see the animal just, pokes its nose out, doesn't want to fall out, doesn't want to fall back, and just stays there for a few minutes. Okay, you might imagine that would be aversive to the animal. Who knows? Well, one way to find out, of course, is you go back, repeat this day after day after day, and you look and see how the animals respond to that, to exposure to the tube. So here now is on the 5th day, the researchers go in and they stick the tube in the environment. And before the video even starts, you can see the animals run into the tube, and you can see actually another mouse competing to get into the tube at the same time. So very simple little trick, essentially, in management, but given that it's something we do all the time, it just shows that sometimes just a little bit of a creative rethink can have a dramatic effect in terms of how, in this case, how aversive the procedure is for the animals. Okay. Close that. Let's go back to our PowerPoint, and I'll just show you, it's actually running on the video there. Go back that. And just show you, here's just one example of some of the effects that this has on the animals themselves after this repeat pairing. And you can see in this case, Hurst and West looked at a variety of different strains of mice, and they're looking at the amount of time that the mice voluntarily interact, in this case, either with the researcher's hand or with the tunnel that I showed you. They used another treatment here, which was the cup. And you can see that in each of the strains, and they're also showing this separately by sex, you see a big difference in terms of how willing the animals are to interact with the restraint device, if you will, when we use these other options. Actually, the cup works almost as well as the tunnel, and votes much better than picking the animals up by the tail. Okay, I give you an example of something that we do to the animals, which the animals at least initially perceive as negative. I have shown that if you use the right approach, we can maybe turn that from negative to at least neutral, and maybe in some cases positive. But I think we could do a much better job in terms of also trying to interact with the animal in ways that the animal perceives as positive. And you might think that this is sort of a radical request when we're talking about rodents, but in fact, if you look at the requirements for the care and housing for some of the other lab animals we work with, this is actually just part of our normal practice. So here is a quote from the guide saying, for dogs and cats, especially when housed individually or in smaller enclosures, should be allowed to exercise and provide with positive human interaction. So maybe not that radical an idea at all. Okay, I'm going to show you one cool video. This is from Jaak Panksepp's work. If you don't know, Jaak, a neurobiologist, has been very interested in affective states in animals, including rodents. And he's done this cool stuff where he has been basically exposing rats to rough and tumble play. He's been recording ultrasonic vocalizations for these animals, and his data indicate that it actually becomes a very positive thing for these animals. The animals will work to have access to a hand. This is Jaak Panksepp's hand that does this rough and tumble play with them. And I'm just going to try, see if I can pull up this tickling video and see if this works okay. Just to show you what this looks like. Here is, so you can see quite a lot of sort of movement from the hand there, and the animal actually coming up and approaching the hand. Now, that's an animal with some training in this, but it's actually a procedure which actually seems to work very well once rats are socialized to the people that provide the tickling stimulus, as Jaak Panksepp calls that. Okay. Let's go back to the PowerPoint here. And just because I'm a scientist, I have to show you some data too. So here is some data from Suzy Cloutier, who's a researcher who works together with Jaak Panksepp. And you can see a bunch of different things here, but here's one example looking at these 50 kilohertz ultrasonic vocalizations. And these are animals looking at in anticipation to some kind of interaction with humans, where either they had minimal interactions or that they were the animals that were tickled. And you can see animals housed in different kinds of ways, but regardless of how they're housed, all those animals are showing much more and any more anticipatory vocalizations, these 50 kilohertz vocalizations, once they learn that they're going to get this rough and tumble tickling event rather than just sort of a minimal handling event. Okay. Let's turn to a little bit more detail in terms of some of the interesting things that Joyce has been doing with her rats in terms of socialization protocols. So here's one of Joyce's Long Evans rats. What Joyce did is right from the very beginning, from when these were pups, she would interact with these animals. She would spend a period of time with them basically every day, gentle, playful handling. She'd provide them food treats. She would get them used to being carried and held, and she would allow them to explore their environment, certainly within the cage, but also outside the cage too, as you'll see in a minute. Here is once the animals are all trained up, here's a video now of the rat, Joyce, interacting with the rats. She's just stuck a rat actually on top of the home cage. Here you see it crawling down the side, and it runs over, it takes a stop and smells the tripod where we got the video camera up for the first time. and it hops into this apparatus that Joyce has built. Joyce was doing some experimental work where she was looking at cognitive tests for these animals, and she wanted the animals to be able to freely enter the tests at their own volition. So she can see in this case the rats are able to come individually down, and they are in this case choosing to enter the apparatus because they know in that apparatus they're going to get some cool treats from Joyce. Go back and, oh, sorry, go back into PowerPoint here, and here's another one just showing some of the daily handling that Joyce did with these animals. So she's picking the animals up, doing some sort of this, again, this rough and tumbling tickling. And if Joyce actually had in her setup, she had just an inexpensive bat detector, a transducer, so that she could hear the ultrasonic vocalizations from these animals when they made it. But you can see all the, in the video, the animals, I'll just go back and start that for a second. The animals actually crawling around Joyce and approaching her for this and handling interactions. The cage door is actually just open as she's doing this. Okay, what kinds of effects does this have on the animals? Well, I showed you already the really, I think, very cool from an experimenter's perspective, effect of having animals which are able to basically provide their consent to participate in the trial because they're voluntarily participating in the experiment. You also have animals, as you can see here, which are very different in terms of how they are to work with and interact with. And so here are these animals interacting, oh, sorry, I've got out of the PowerPoint here. Here are these animals interacting with Joyce, but it's not just Joyce that they that the animals are highly interactive with. So here, for example, is, just go forward one video. And this is now Joanna. And this is, I think, the very first time Joanna has interacted with these, right, these rats. And you can see Joyce's rats just crawling all over Joanna and not being sort of fearful at all in terms of their interactions. Here's another animal just approaching Joe. So it gives you a sense of what the animals are like to work with when they have gone through this type of socialization protocol. These are important to distinguish, not just animals that are simply habituated, that is to say, willing to have us approach and pick them up, but animals that are actually socialized, and as you can see in this case, seeking out and responding positively to human interaction. Here's another researcher in the lab and just doing some sort of cage husbandry work, and the rat climbing up and interacting with them. And you can see in a way that at least he perceives to be as positive. For people that aren't so used to working with these socialized rats, might be sort of a little bit scary the first time, but I can assure you that it becomes very pleasant to work with these animals. Okay, so let's just have a bit of a rethink then in terms of what are the things that we could be doing in terms of our socialization protocols for working with laboratory animals. Current procedures, we focus on habituating. So if we're going to do a procedure on a good lab of the animals, we will be repeatedly exposed to the procedure, let's say for a number of days before we start collecting data. So we begin to minimize the distress response that the animals show, but we're not attempting to socialize the animals. If we do socialize the animals, then we get animals, in this case rats, that really seem to enjoy human interaction. And as I've argued at several points so far this morning, that I think that we also benefit from having these interactions with animals that are much more pleasant to work with and actually animals that are interested in working with us. Joyce says that these socialization schemes are actually easy to implement. And if you think about it, I mean, it's not something, it requires time, but it isn't something which necessarily requires a great deal of investment in terms of infrastructure. In fact, at UBC right now, we're starting to implement it in part of our main veterinary facility where we train students that come to the university and that are going to be animal researchers, where we train them to interact with rats. We are now implementing these same procedures so that those researchers will have as their first experience in terms of working with animals these much more interesting interactive animals. And what we found is actually it's very easy to attract volunteers to come in and do socialization work with these animals. It's actually a very pleasant task for students to be engaged in. What's very important, though, is that the socialization begins early in life. Both Joanna and Joyce's studies, we started when rats, they were brought in at weaning, and it's actually better to start before weaning if we can. So here you can see, in fact, Joyce interacting with her rats on the very first occasion. You can see the rats are just a little bit, well, more than a little bit, are unwilling to approach Joyce's hand when they first see it. And it takes a chunk of time before the animals become interested in interacting with Joyce. In contrast, here are some Long Evans rats that are kept with at home as pets and have been interacted with daily in the pre-weaning phase. You can see the animals are much more interactive and interested and less fearful in terms of interacting with the human handler. So some of the tips that if you talk to people who keep rats as pets will tell you, if you want these sort of well-socialized animals, is, as I said, handle earlier, ideally before weaning. They also actually suggest weaning a little bit later in terms of having animals which are nicer to work with. And of course, one of the really interesting things that the pet community are doing that we haven't taken advantage of, I don't think of properly in the lab animal environment, is actually breeding for temperament. If we really care about having animals that are pleasant to work with, we need to be working with the breeders so that we get animals that are pleasant to work with. I mean, it's not rocket science, but it really is a very important factor that I think, again, we could be making really rapid progress on if we start asking our animal breeders to provide us individuals which have the right temperament for this kind of positive social interaction with humans. Another, I guess one of maybe the last point I have here is that we need to also, I think, be thinking about what happens to these animals after we're finished with them in the lab. Of course, typical fate for laboratory rodents is euthanasia. In our group, we've done a lot of work with euthanasia. Take-home message from that is there are much better ways that we can be killing rats rather than CO2, which we typically use. I won't get into that now, but of course, the best thing we could do is be finding another home for animals. And in a lot of the good facilities that are working with, let's say, laboratory dogs, this is really just internalized as part of the way that we do business. Is the idea is that the animals are well socialized and brought up as somebody's future pets. They're used now for research, but we want animals that ultimately be adoptable and will make good pets. And I think that's one of the really interesting features of what Joyce has been doing in these experiments is she has rats which are super interactive and interesting to work with. And once Joyce was finished with her experiments, these rats were actually ridiculously easy to adopt out. And so at least I think this is a conversation we need to start thinking about when we come to all the animals we have in laboratories is what's our role as stewards of someone's pet, someone's future pet, and does that make us rethink what our responsibilities are to those animals? Anyways, an interesting area for you to think about. Okay, quickly wrap up now because we're running out of time. A few things that I want you to think about. I know I've done a lot of blue sky thinking about where we might want to go in the long run, but there's also some short-term things that I think you can think about. One is just at the very sort of what I call level 1 type interventions are the idea of adding interactive treats. This is something that can be done even in conventional caging. I've given you a few different examples of what we can do. This idea of using gentle handling procedures, including tickling for the rats and those tubes for picking up mice. For those that are a little bit more ambitious, I think there's some things that you can start thinking about. The idea of providing even standard cages with raised lids allows the animals to do some interesting and important behaviors. This opportunity to implement a socialization protocol, of course, is possible even for animals in standard caging. So we could be making the lives of these animals much more interesting in terms of our interactions with them, even if we don't change the caging system. And the added advantage of these kinds of socialization protocols is I think they also at least allow an interesting discussion about an adoption policy for eligible animals. And in the longer term, of course, I really want you to be thinking about using examples of having housing for these animals that allows them to show their full range of behaviors. And it comes back to what I said we're doing now in our current animal facility here at UBC is having people's first interactions with animals to be these much more sort of interesting, interactive animals. And with the idea of really changing the way our new researchers that come into facilities think about our rodents and therefore their responsibilities to these animals. The idea of adding routine socialization activities which include positive interactions. And I guess my last point is that these are just examples that Joyce and Joanna have done in terms of trying out some neat things with these animals, but I really encourage you to bring your own creativity to bear in terms of ways that we can be improving the housing, but also socializations for our lab animals. And with that, I'd like to thank you very much for your attention, and also thank the sponsors for the work that I showed you today.
I think there is some time for questions. Anyone from our audience or online? I have a question for you. Given how inactive the rats in standard caging look compared to the ones in enriched caging, do you have a comment on either transitioning animals from the old-fashioned housing to novel housing and how it might affect the validity of tests, you know, the comparability of data derived in the old-fashioned housing? And conversely, do you have a reason to question the validity of some of the models using animals in boarding housing?
Yeah, I mean, I think to me that's the sort of sobering thing. You look at those two videos and you ask, well, which is the real rat? And does it be like, you know, anyways, it's easy for me to, obviously, as you could tell, I am passionate and interested in this area. But to me, I look at those rats and those standard housing, and what can these rats, and this isn't even a naturalistic, right? This is just, you know, a reasonable caging that's provided by some of the sort of the rat fancier communities. So this is still nowhere near as complex the kind of environment that you would see in a wild or a semi-naturalistic situation. And our conventional cage rats are just nothing like these animals. So I don't know what those rats could possibly tell us about real rats. Sorry, it's a bit of a brutal assessment, but that's sort of what it looks like on my side. So I will say that Joanna Mikowska is just in the final stages of her PhD, sort of capturing a bunch of data on these animals, and so the sciencey bits, the data from those animals is still to come. The observations, sort of my own observations, and hopefully Joanna would agree with me on this, is that though animals in the enriched cages are fitter, they're able to do much more, and that plays out in terms of some of the health outcomes we see from the animals in the two different environments. But I better not say much more than that, or else I'll probably get in trouble with Joanna.
So the question was whether adverse procedures would affect, if you've socialized a rat, whether if you also had to do an adverse procedure, whether it would affect the relationship?
So a very interesting question. I'd almost turn that around and ask, I mean, as a lab community, for us to be looking more at places like zoo environments, for example, where you have animals. I mean, I've got this great video, for example, of this elephant receiving some foot care in a zoo. And I do a lot of work on dairy cattle, and our standard way of doing foot care on dairy cattle is sort of basically stick it into a crush and pull up its leg and sort of use a bunch of mechanical force to get access to the limb and then do what we need to do. Of course, that's very difficult, if not impossible to do in most elephant facilities. And so what they've had to use is a series of positive reinforcements where the animals are trained to provide its foot to the technician so the technician can do the care. Even actually sometimes quite painful procedures, the animal is choosing to allow the operator to do that because it knows that there's going to be something good at the end. And I've seen the same thing actually for monkeys sticking their arm out of a cage to provide a blood sample, et cetera, et cetera. And I think actually this really comes down to, in terms of probably what it means even to suffer for us or for animals under our care has a lot to do with our perception of control, probably as much or more to do with our perception of control than it does to do with the immediate pain associated with the procedure. So you and I, all of us do things all the time which causes some short-term pain, but we do it willingly because we believe there's some kind of positive reward at the end. And we do it under our control. And that's very different if somebody was to force us to undergo the same procedure that we didn't understand and that we could not control. And so I actually think there's a lot more we could be doing with our lab animals where we trained them basically for positive reinforcement to take part in procedures, sometimes maybe even unpleasant procedures. I don't know how far that could go, but I think we could go a lot further than where we are today.
Okay, thanks very much. I think we have to wrap up. I neglected to thank the Lawrence Foundation and the Tompkins Foundation for helping to sponsor the work today. Thanks again, Dr. Weary.
Oh, my great pleasure. Thank you for your